Good afternoon. Welcome, everyone. If you were with me yesterday, welcome back. If you're just tuning in today, no worries. This is part of a two-part series, but you can certainly join on part two. Yesterday was content for TEAS scientific reasoning, and it was all just content to prepare for today. So today is our practice test based on the content we had yesterday. However, if you weren't here yesterday, still stick around. This will help inform you as to where you're at on these topics, and then you can obviously go back and see some of our old free webinars, or you can take a look at our on-demand videos to get caught up in any of your areas that you're not as strong in. So please stick around. Today, I want to first just kick off with going over some of the things we have to offer for you here at Archer Review. And by the way, I am Shellie Harshberger coming at you again, or for the first time if you weren't with me yesterday. So I'm so pleased to work for a company that has so many resources for you to help when you're studying for a big exam. I have spent many, many years in school, and I know how stressful it can be to prepare for those big exams. And so I just feel fortunate that we have so many wonderful things to offer. So we've got QR codes on here. If you have an iPhone, you can take a screenshot and those QR codes will activate. Otherwise, you could still take a screenshot with an Android and access via the addresses that we have on here. But I wanted to point out, yesterday I went over our question bank. Our question bank is pretty spectacular. It has over 2,000 questions covering all the topics of TEAS. And you get the setup, the question looks very similar to the TEAS, and you can get immediate feedback on your answer choice with rationales, and you even get linked on-demand videos. So since I went over the question bank yesterday, I promised everybody that I would go over the on-demand videos today because those are pretty spectacular as well. So let's take a look at those. They're just really downright amazing, to be honest with you. So our on-demand videos, that's what I've got on our screen here. You can see I'm toggled into on-demand, and we have English, reading, math, and science. So we cover all of the topics. So since we're in science and since we're in scientific reasoning, I wanted to go to the scientific reasoning, the first one. So there are a couple of things I want to show you here. First of all, the video is not, it's not too bad. It's about 20 minutes long, 20-minute stint. But let's say you really only needed to go over dimensional analysis. Well, you could go right to that section right here. That's what the timestamps are. And it's only 51 seconds long. Or maybe you wanted to do some practice questions that are guided. That one you could go right here and it's only a minute long. And so that's one of the really cool things that I wanted to show you. The other thing that I wanted to show you was the fact that we not only have the videos for you, but we also have handouts and cheat sheets. So here, for example, would be our scientific measurements tools cheat sheet. And I love these cheat sheets. These are just so cool. I mean, check this out. This gives you great visuals of what base units are used in the metric system, some conversion tips, our conversion chart, some example problems, tools that we would use, and key principles. So I love our cheat sheets. I think those are just so neat. And then the handout. The handout is the slide set. So a lot of times I've heard students say that they will print out the handout and then write notes on it. They'll annotate on it as they're going through the on-demand videos, which is a really cool way to make yourself like actively involved in that video. And even if you don't have the ability to print them out, if you have a couple screens, you could use this on one screen to do some of your own annotating electronically as well. So it's just really, really neat options we have for you there. And I wanted to make sure everybody got to see that. So additionally, as you probably already know, we do offer tutoring. We offer all subjects. And here, let me show you what your tutors look like. Oops, went too far. So there are your tutors. So Tammy is our English and reading specialist. As you know, I'm Shellie Harshberger. I'm your science person. So I can help you with chemistry, biology, anatomy and physiology, and obviously scientific reasoning. And that's what we're learning about today. And then Brandon Math has all of TEAS. Brandon Craft has all of TEAS math covered. So you can scan those QR codes, but those are your tutor options. Furthermore, we have, as you know, free live webinars. We have many webinars every month. Just myself, I put on 6 webinars a month, 4 biology and 2 scientific reasoning. And I know we have right around that for reading, right around that for English and anatomy and physiology and math. So we have tons of free webinars for you. You can also rewatch old webinars by using that QR code. It's not every old webinar, but it's quite a few of our past webinars. And then, of course, just to give you an opportunity, we do have a couple bundles. We have the SurePass and the SurePass Pro. The difference here, the SurePass, you get everything but an hour of tutoring. And with the SurePass Pro, you get that hour of tutoring. And it can be in any subject. It doesn't just have to be in scientific reasoning because you're coming at me, or I'm coming at you with that today. So it can be in any subject. So definitely take advantage of those. Also, as you're probably fully aware, we are all over social. So TikTok, Insta, Facebook, and not only are we present there, but we put out 2 practice questions and 2 content kind of overviews every week. And we generally feature a topic, like this week is math. And so you get some help that way too. We'll have a practice question. We'll go over the rationale. We'll have little cheat sheets and little topics to cover as well. So definitely follow us on all the socials and you'll get that added help. And that's all for free. So that's pretty exciting. Last but not least, before we dive in today, if you are in need of tutoring and maybe the schedule just doesn't seem to align with yours, definitely email us at prenursing@archerreview.com. Sometimes we can move our schedule around to accommodate you. And we have certainly done that in the past for many people. So feel free to reach out and we can try to get you booked. Okay, so let's talk about the format for today. This is set up like an actual practice test. So I will reveal the question. I'll actually read the question, but not the answer choices. I've got feedback in the past that it's actually distracting when I read too much of it. And I will set a timer on the screen for 45 seconds. What I'd like to ask you to do is look through that question, look through those answer choices, and however you're coming at me, put your answers out there in the chat box, whether it's Restream or whether it's YouTube or Instagram or Zoom, put those answers in the chat box. That helps you be actively engaged, and the more senses you activate, the better you remember things. Now, just as a full disclosure, if you signed up via Zoom, I am the only one who sees your answers. So you get that little added privacy. If you are coming at me on any of the other socials, everyone does see your response, but I still strongly encourage you to participate because the more active you are, the better that you'll remember. Okay. All right. Without further ado, then we are going to dive right in. Our first category is scientific measurement. Which of the following correctly orders these metric prefixes from smallest to largest? Get those answers out there. All right. So our correct answer is B. Okay. From smallest to largest, it goes nano, micro, milli, deci, kilo, based on the list you were given. Now, do remember that we have that silly phrase, now many millipedes crawl down mountains, to help you remember all of them. And so that would be an option to help you get through, at least up to the base meter. And then you would have had to have known that kilo was beyond meter, but that's a start. Now let's take a look at the other ones. Answer A. Answer A, the problem was that milli should become before deci. Okay, and so in answer A, we started with deci, and that is a problem. We have to have milli come before it. Okay, all right, looking at answer C, milli is incorrectly placed after kilo. So if we look here, we've got milli after kilo, and so that's a problem. And then answer D, nano is smaller than milli. And this problem reverses them. So that again puts it in the incorrect order. All right, folks, let's move right along to our next one. A student is measuring the volume of a liquid in a graduated cylinder. To obtain an accurate reading, where should the student measure the liquid's volume? All right, are we ready for our reveal? Our correct answer is B, to measure at the lowest point of the meniscus. So if you remember from yesterday's content, we want to get right in here at that lowest point of the meniscus. Now I can see that C could be confusing. The option for C was at the middle of the liquid's curve. And I think the way that that one's worded can be confusing. And, you know, we'd like to talk about those confusing ones because those are the ones that can mess you up on the TEAS. And so we want to be focused on the fact that it's not the middle of the liquid's curve, but it's the lowest point of the meniscus. So when you're looking at that arc of the meniscus, you want to focus on the very lowest point. And I think that that is the critical component there. But I could see why C would cause some confusion, and I like to talk about those confusing ones. So now as far as our other answers, at the highest point of the liquid for A, no, measuring it at the highest point of the liquid is going to really overestimate that volume of the liquid. Our next one then, D, at the top of the container. Well, that one's really not logical because you don't want to measure it at the top of the container. Like our example right here, if we measured it right here, what does that tell us? That tells us how much the container holds, but it doesn't tell us where the liquid goes to in the container. And we're trying to measure the volume of the liquid. So that would be illogical. And that's one of the things that's really important with scientific reasoning, is to keep in mind that a lot of scientific reasoning is really about just using logic, stopping and thinking, does that make sense? I used to have a chemistry professor who would tell me to put my common sense hat on because, yeah, those are the kind of errors that we make. And then we kind of get disappointed with ourselves, like, why did I do that? I knew better. So certainly make sure you're applying that common sense, or as my professor would have said, putting my common sense hat on. All right, folks, let's take a look at our next one. A nurse needs to administer 1 gram of a medication to a patient. However, the pharmacy only provides the medication in 250 milligram tablets. How many tablets should the nurse administer? I saw some brave souls who did answer, and then some who weren't so brave and were like, oh, I'm going to hold off on this one. So let's take a look and reveal our correct answer. Our correct answer is C. So we have to do some math here. We have to, first of all, remember that gram is our base unit, and that 1 gram equals 1,000 milligrams. Okay, then each tablet contains 250 milligrams. So the number of tablets needed needs to equal 1,000 milligrams. So we divide that by 250 milligram per tablet, and we realize that we need 4. Okay. Or even if you just kind of thought it in your head, okay, they're 250. So 1 tablet is 250, 2 tablets are 500, 3 tablets are 750, 4 tablets, there you go, there's the 1,000. And the 1,000 milligrams equals 1 gram. Okay. So our other answers, answer A was 2 tablets, but remember 1 tablet's 250, so 2 tablets would only be 500. Answer B was 3 tablets. Well, if they're 250 each, 250 times 3 is 750. So again, that would not be enough. Answer D, 5 tablets. Well, 4 equals 1,000 milligrams. And if we add 1 more tablet, that would be 1,250. So that would be too many. So again, I challenge you to really think about this logically. The big thing you needed to know to help, get to the answer was that 1 gram equals 1,000 milligrams. And then you could start thinking about logic. You could even, sometimes I like to transfer it to money in my brain and think, okay, if I have $1,000, what percentage of my 1,000 is 250? And I could kind of go backwards out of it that way too. So there are many ways to come up with that answer, but that is our correct answer. Okay, folks, let's take a look at our next category. Remember, this is interpreting and evaluating scientific information all combined into one. So keep up with me here, folks. We're doing good. I'm seeing a lot of good answers out there, and I see people taking chances, and I love that. That's how we learn. All right, which of the following are characteristics of empirical evidence? Select all that apply. Oh, I saw some good answers out there, and I saw some really close answers. So let's break this down. Our correct answers, I'll first reveal, and then we'll go over them, are A, C, and E. And I saw a lot of people who are really close on these. So let's really break this down. So A, based on direct observation or experimentation. Now, remember that science as a whole bases its entire understanding on empirical evidence. Okay, empirical evidence is evidence derived from experiments or direct observation. So that's kind of the meaning of empirical evidence. Then C was our next correct one. Remember that it needs to be measurable and repeatable. So we have to have data, something that we can measure so that someone else could come along and repeat that experiment and potentially have the same results. Okay, finally, E, collected using systemic methods. Now, remember that I talked about even the TEAS book will kind of do the observation, hypothesis, experiment, and they'll do it kind of linear. And not all experiments are this perfect scientific method line that we learned in elementary, but it does happen in a very systematic way where someone else could come along, follow that system, and have the potential to have the same outcome. All right, let's take a look at our two incorrect answers then. What's wrong with B? Primarily subjective and influenced by personal opinions. Remember that that really speaks to something we learned about yesterday, bias. Okay. And we don't want bias in a scientific experiment. There's no place for it. So we want to try to avoid being subjective. We want to avoid that. And we certainly want to try to keep our personal opinions out of our scientific experiment. There are some schools of thought that say we can't, we cannot fully 100% keep our personal opinions or our subjectivity out of our scientific experiments, but we want to try very hard doing so. All right, D, what's wrong with D? Derived from logical reasoning alone. The key word here that makes this so incorrect is alone. Now, scientists do apply logical reasoning and they do make judgment calls. And that's part of why we have to make sure that an experiment is repeatable, because what one scientist's judgment call is may not be the same judgment call another scientist makes. And so there is this idea of logical reasoning that does have to be applied, but it isn't the only characteristic. That word alone is our issue. So well done. I hope that explained it for the few of you that had a couple of them incorrect. Let's move right along. All right, in an experiment, what is the purpose of the independent variable? Okay, are we ready to reveal our response? The correct answer is C. That's what's being deliberately manipulated. So let's take a look. It's intentionally changed to observe its effect. So remember, the independent variable is the factor that we're really experimenting, that we're trying to figure out. So, for example, yesterday we talked about like migraine medication. If we wanted to try a new migraine medication on two groups of people that suffer from migraines, one group would get the migraine medication, and that would be the independent variable. It's what's being changed to observe. Does it decrease migraines for people? Okay. All right, so that was C. That was our correct answer. Now let's take a look at our other responses. A, it remains constant throughout the experiment. While we do have controlled variables that do have to remain constant, if we stick with the migraine example, we would want all of our migraine participants to keep up their regular sleep schedule, their regular diet, their regular exercise regimen, so that we could know for sure that it was only the migraine medication and not something else they changed in their life. So there are constants, but they are not the independent variable. B, it is the factor that is measured in response to changes. That's actually the dependent variable. In our migraine medication example, we would be measuring if our migraine patients suffered less migraines or less severe migraines, and those results would be our dependent variable. D, it serves as a baseline for comparison. Remember, the baseline for comparison is actually the control group. If we stick with the migraine example, we would have two groups of migraine sufferers. One group would receive the new migraine medication, and the other group would either receive no medication at all, or they would receive a placebo. Remember, a placebo is like fake pill, a sugar pill, so that psychological expectations don't influence the results. Okay, you guys are doing great. I'm seeing good answers out there. You're staying nice and attentive and active. Thank you for that. Well done. Let's keep going. Which of the following best describes the purpose of a control group in an experiment? All right, let's reveal our answer. Our correct answer is B. Let's take a look. To serve as a baseline for comparison. Remember the control group, if we stick with our migraine example, they're not getting the medication. They're our baseline. That way we can see, did the medication decrease severity or decrease number of migraine headaches? And the baseline would be they should be having their headaches like normal. That's our baseline for comparison, our control group. All right, let's take a look at our other choices. A, to receive the treatment being tested. That is our experimental group. So that's the group that's receiving the treatment. That's our experimental group. C, to ensure that all variables remain constant. We definitely want our variables constant. Sticking with our migraine example, we don't want them to change a thing different from what they normally do. We want them to stick with their same diet, their same sleep, their same work schedule, their same everything, so that we can attribute any benefits to the medication. D, to intentionally manipulate the results. We never want to intentionally manipulate results. That would be unethical, that would be inaccurate, and that would not be serve any purpose whatsoever. So we certainly wouldn't want to do anything like that. All right, folks, let's check out our next one. What is the function of a placebo in medical trials? Okay, I want you to pat yourselves on the back because from what I see, I think everybody who put in an answer had the correct answer. So you ready to see that? You want to pat yourself on the back? The correct answer is C. So it's a harmless, inactive substance given to that control group to assess the true effect of the actual treatment. So this was our correct answer, and I think everybody got that. So very well done. So A, to provide an alternate form of treatment. No, if we provide an alternate form of treatment, then we have no baseline to compare the medication we're trying to learn about to. B, to ensure every participant receives active medication. No, unfortunately, when people sign up for clinical trials, they know that they may not actually get the active medication. It's an unfortunate situation, but a very needed one to assess the effectiveness of that medication. And then D, to measure how different independent variables interact. There should only ever be one independent variable in our experiment. That way we know it is the only thing causing an impact on the dependent variable. So there should only ever be one independent variable. All right. Which of the following are important factors when analyzing the design of an experiment? Select all that apply. We ready for our answer reveal? Here comes. A, B, D. So let's take a look. A, ensuring a large sample size and repeated trials for reliability. Absolutely. We talked about yesterday, if our sample size is too small, it doesn't provide a sample of the population. It's too small. Maybe we have all of one gender, or maybe we have all of one race or one age group or something along those lines, or too many of the same. And so we have to have this heterogeneous mix of people to truly assess, in this case of if we're sticking with the migraine medication, to truly assess the migraine's medication's effect. So we have to have a large sample size that kind of accurately represents the population. And then repeated trials. The more times the experiment has been done with the same results, the more confidence we can have in that experimental design. Okay, our next answer, so that was A. Our next answer was B, that was correct. Keeping all conditions except the independent variable constant. This is so, so, so important. And this one was the one I gave the example with yesterday, that not necessarily a migraine sufferer, but let's say you have a headache and you say, I think maybe I'm dehydrated. I'm going to drink, a half a gallon of water today. And then halfway through the day, you're drinking your water, but your headache's really bothering you and you take Tylenol. And by the end of the day, you're like, wow, I'm feeling pretty good. My headache's gone. Well, is it because you drank more water? Or is it because you drank or because you took Tylenol? You don't know. Because now we've got two independent variables, we've got two things we've changed, and we don't know which one to rely on. So that's why A, we have to have one independent variable, and then B, we have to keep all other conditions constant to see which actually got rid of our headache. Was it drinking more water or was it taking the Tylenol? Okay, and then D was our other correct answer, maintaining control variables across all groups. And that's really hard when you're dealing with humans. If we're going to go back to our migraine example, we have to tell these migraine sufferers, you have to live life just like you normally would. Don't suddenly decide to drink more water, get more sleep, or do any of these other factors that are different than your usual day to day, because that will influence the outcome of our experiment. And that's a hard thing to get humans to do, but it's true. All right, so let's see what's wrong with C and E. C, allowing participants to choose whether they receive the treatment or placebo. So the whole idea of placebo is so that we don't have a psychological impact, so that we don't just feel better because we think we should feel better because we're taking new medicine. So if we get to choose, that influences that psychological impact. So that's what makes that incorrect. All right, let's take a look at E. Changing multiple variables at once to see various effects. And that kind of speaks on what I was saying about if you're dehydrated, you're drinking water, you're taking Tylenol, you don't know what's getting rid of your headache. You've got too many variables that you're changing. And so that's what makes that incorrect as well. All right, let's see what our next one is. What is the primary purpose of a double-blind study? I saw a few more people just joined. Welcome. All right, are we ready to see our answer? I think we got to do some patting ourselves on the back here. If you chose A, congrats, and I think many of you did, so very well done. So our double-blind study is that we want neither the participants nor the researchers to know who's getting that treatment, okay? That way, expectations from the researchers can't influence how they perceive results, any more than the patients can influence the outcome based on what they perceive as results. So it ends up being completely, well, I can't say completely unbiased because, again, there are some schools of thought who believe that even the tiniest bit of bias creeps into every experiment. But I would say it's the least biased approach possible. All right, so A was our correct answer. So let's take a look at what's wrong with the rest of them. B, to allow researchers to decide which participants receive the treatment. So that's essentially the opposite of what we want to do, right? We don't want the researchers to influence which participants receive the treatment. Because let's say, for example, this is a migraine medication company, and they're doing their own independent research, and the researchers are going to get a bonus if their medication works. Do you think that's going to influence how they perceive results? Maybe, right? So we certainly don't want researchers deciding who receives the treatment, okay? All right, C, to test multiple variables at the same time for efficiency. So we've talked about that quite a bit. If we have more than one variable changing, we don't know which one is responsible for the outcome. So we can only change one variable at a time. D, to guarantee that every participant receives some form of treatment. And we talked about that earlier too, that no, every participant is not going to receive some form of treatment. That is all part of having a control group and an experimental group. Unfortunate, I'm sure in some cases, but a necessary evil if we want to determine how effective that medication is. All right. Which of the following are important guidelines for recognizing bias in an experimental design? Select all that apply. All right, I saw some good answers out there. Let's see how we did. A, B, and D. So these are correct answers. A, B, and D. So let's break them down. A, ensuring participants are randomly assigned to avoid skewed comparisons. Absolutely. We want participants to be randomly assigned because, again, we don't want that factor of bias. If we really have our heart and soul invested in this new medication working, we might be even subconsciously focused on trying to choose the young, athletic, healthier version of participants in order to have the best results. And so that would be an issue. So we couldn't do that. We want to make sure that they are randomly assigned. B, using blinding, single or double, to reduce bias from expectations. Yes, remember, double blinding is both researchers and participants don't know who's receiving the treatment, and single is participants don't know who's receiving it. The most secure form, or the least biased form, would be double blinding, where no one knows. That would be the best. All right, D, being aware of potential conflicts of interest or funding sources that could influence results. And I talked about that yesterday, and I've inferred that several times today. We have to really be careful because if the company that created the medication is also the company doing the clinical trials, there's a conflict of interest there. And it happens, right? It's when we get a third-party study, when we get an independent study that really gives us confidence in that experimental design. All right, so let's take a look at C and E. C, allowing researchers to selectively assign participants to groups. So that goes along with A. We certainly want them randomly assigned because we don't want those researchers choosing who they perceive as the healthiest for their group that's going to receive the treatment. So that makes C incorrect. Now E, choosing participants based on expected outcomes to confirm hypotheses. No, that would be like the master of bias, right? So now we're like trying to pick the perfect participants to make sure that they support our hypothesis that our medication works. We certainly do not want that either. All right, we got one here about a particular study. Let's take a look. A researcher is studying the effect of a new allergy medication on symptom severity. One group of patients takes the new medication, while another group does not. After 2 weeks, the researcher records the severity of allergy symptoms in each participant. Which of the following correctly identifies the independent and dependent variables? So that's what we're looking for in the experiment. All right, we ready for the big reveal? Here goes. Correct answer is B. B is our correct answer. So let's take a look. Independent variable, the administration of the medication. So the medication being given to the group. The dependent variable, their allergy symptoms. Okay, so it's the group that's getting the medication. The medication is that independent variable. Dependent variable is what their symptoms are like. Okay, now let's take a look at our other answer choices. A, independent variable, allergy symptom severity. That's the dependent variable. It depends on that medication that we're giving, right? And that's one of the ways that you can remember which is which. The dependent variable is depending on that thing you're changing. We wouldn't have any need to assess their allergy symptom severity if we didn't do something to change the potential for them to have less severe allergies. Okay. So then dependent variable, whether or not the patient takes the medication. That's just ensuring that the patient does what they're supposed to do, but that's not one of the variables in the experiment per se. That just makes sure that they follow their expectations. All right, C, independent variable, time of day the medication is taken. If we look back at our passage, there is no discussion about time of day whatsoever. This is not discussed. Okay, so if it's not mentioned in our passage, that's not going to be a critical component of it. Dependent variable, type of allergy medication used. So type of allergy medication used, you might have been able to kind of filter that into part of the independent variable, but it's certainly not part of the dependent variable. The dependent variable is the outcome of receiving allergy medication. All right, D, independent variable, pre-existing allergy conditions. There was, again, no discussion of that in the passage. And that's not what we're measuring. We're not looking at whether they had pre-existing allergy conditions. We're looking at the effectiveness of an allergy medication. And so then dependent variable, dosage of medication given, again, that was not discussed in our passage at all. So if it's not discussed in our passage at all, it's not going to be one of our answer choices. And that's kind of a nice little test strategy. In a situation like this with an experiment, if they're not even discussing it in the passage, it's certainly not going to be one of the variables. All right, we are moving right along here. We are on our last category, predicting relationships. Let's take a look at our first question. Which of the following lists the levels of biological organization in the correct order from smallest to largest? Okay, our correct answer is C. And it was atom makes up molecules. So like that molecule of water is made of 2 atoms of hydrogen and 1 atom of oxygen. Then molecules make up organelle. So we think about when we talk about like macromolecules, carbohydrates, proteins, lipids, things like that. Those would be the types of molecules that are going to make up the structures within our cells, such as organelle. Remember that organelles are just like tiny little organs within our cell. So of course, they're going to be smaller than the cell. Then cells make up tissue. Then tissues make up our organs make up our organ system, and then our organ systems collectively work together to make up the organism. So now let's look back at our other answer choices and see what went wrong. So A, we start with molecule and then atom. So before I go any further, I realize, well, there's a problem right there, okay? Because atoms make up molecules, not the other way around, okay? Otherwise, that one would have been correct. From here forward, it's in good shape, but it's got molecule and atom out of order. All right, our next one, our next B, atom makes up molecules. So far, so good. Oh, but then we have cell and organelle. These two are out of order. Organelle are like organs inside the cell, so they're smaller. Then from there forward, it looks pretty good. It's correct from that point on. All right, C, we know this was our correct answer. Let's take a look at D. D, molecule, atom. Oh, see, those two are out of order. Oh, cell and organelle are out of order as well. And then looks pretty good from there on. Okay. How does the sequence of events in a biological process demonstrate a cause and effect relationship? How'd you do? Our correct answer is B. Let's take a look. B. So earlier events directly influence or determine the outcome of later events. That's a cause and effect relationship. And if you're familiar with cause and effect relationships, even if it's not in a biological process, you can apply that logic again that something causes an effect, right? So something early causes something to happen later. All right, let's take a look at our other answer choices. A, each event happens randomly without influencing the next step. If that was the case, there would be no relationship, right? If everything was happening randomly, there wouldn't be a relationship. And so that's what makes A incorrect. Now, C, later events influence earlier events in the sequence. I love this answer choice because it goes back to this idea of really applying logic. And sometimes when you have a test that means so much to you, anxiety can kind of take place and you'll, maybe choose an answer that later you go, oh, why did I choose that? And this is one of those. How do later events influence earlier events if we don't have a time machine, right? But I could see how if we're anxious and nervous about this test that means so much to us, we could accidentally choose, an illogical answer. So certainly work on ruling out those illogical answers. And especially too, this is one test strategy. When you first sit down to take the TEAS and you're all nervous and you're trying to get situated and comfortable and, and you got those nerves, flag your early questions to come back to if you have time. Because then once you get in the swing of the test, Most of the time, some of those nervous jitters kind of settle in. They settle down a little bit, and you can go back and find these illogical responses that you might have accidentally chose. So that's a nice little test tip. All right, D, all events happen simultaneously with no connection between them. Again, this idea of a relationship doesn't go with this idea of no connection between them. And that's what is the situation there. Okay. All right. So I got a couple questions. So someone asks, is earlier event always the cause in a cause and effect relationship? Um, in a sequence of events in a biological process, I would say 99.9%. Especially in biology, we like to never say 100% because there's always some exemption to a rule, but I would say 99% of the time, yes. And I also see that I have a question that goes back to this illustration here. So let's talk about this. Our person asked about how like organelles come from molecules. Okay. Now remember that when we study biology, which we do have free biology webinars, we have 4 primary molecules that are both in us and several that we eat. And that would be things like lipids, carbohydrates, proteins, amino acids. And so those molecules then make up structures like the organelle. Okay. And that's the idea there. So while this is a visual of water, we also have those macromolecules. And so I would highly recommend you take a look at our free biology webinar to help clarify that idea a little bit more, but that helps answer the question for today. All right, here's another one. Which of the following correctly describes the sequence of events in a feedback loop? Ready for that correct answer? I see a lot of you have it already, so very well done. Our correct answer is B. Okay, so our order is that we have that initial stimulus that causes some sort of a variable change. That's what we see right here, which then activates receptors. And those receptors send a signal to our control center. The control center then sends signal out to our effectors that will then influence that variable change. All right, let's look back and see what was wrong with our other answer choices. So B was our correct answer. A, control center evaluates the change. Well, I know before I go any farther with A that that's incorrect because we didn't even see a change yet. The control center is evaluating something that hasn't happened yet. And so with that in mind, we can rule out A instantly. Okay, because that is certainly not going to, it's not going to evaluate something that hasn't happened yet. All right, let's take a look at C. Effectors alter the variable. Well, we haven't even encountered a variable yet. So again, right out of the gate, before I read any more of that, we can rule that out because we haven't even encountered a variable. D, receptors detect change. Well, we haven't had the stimulus to generate the change, and we haven't had a variable or the change. So even right out of the gate, I can rule that one out as well. All right, you guys are doing good. We have 2 more questions left. You guys are hanging in, doing such a good job. Thank you for that. Let's take a look at our next question. Which of the following statements correctly describe positive and negative feedback loops? Select all that apply. I'm seeing some good answers out there. You ready for the big reveal? All right, let's take a look. A, B, E, and nicely done. I've seen several people with A, B, E as the answer choice. So well done. A, B, E are our correct answers. So let's take a look at them. Negative feedback loops work to restore stability by reversing a change in a variable. Yes, remember that negative feedback loops tie that word homeostasis to negative feedback. Okay, that's a key word there. Tie those two together in your brain. B, positive feedback loops enhance or amplify a change until a specific outcome is reached. Yes, tie positive feedback loops and amplify together in your brain. Very well done. E, negative feedback loops help regulate body temperature, blood sugar levels, and blood pressure. Yes, and negative feedback loops really regulate everything that needs to be balanced within our body. Okay, that's really the bottom line there. All right, let's take a look at what's wrong with C and D. Negative feedback loops push the body farther away from homeostasis. That is the complete opposite of a negative feedback loop. Negative feedback loop's goal is homeostasis. So that is the opposite of it. D, positive feedback loops are more common in maintaining daily physiological balance. No, positive feedback loops are for specific circumstances. Childbirth, you've got a cut and we need blood clotting, things like that. These are very unique, specific circumstances, not day-to-day maintenance. Our last one, you guys ready? Here we go. Which of the following scenarios is an example of a negative feedback mechanism? Ready for the reveal? Correct answer is B. Okay, so remember, negative feedback, B. When body temperature rises, sweat glands activate to cool the body down. Remember, negative feedback works kind of like our AC or our furnace does. We have a range, we have a temperature we're trying to keep it at. In the case of our body, we're trying to be at 37 degrees Celsius or 98.6 degrees Fahrenheit. And so when that body temperature rises, then the negative feedback is going to try to bring it back to balance by activating sweat glands. If we were to get too cold, our body temperature would get too cool. We would have shivering. We would have blood vessels constricting to try to get our body temperature back to that set range. All right. Now, A, C, and D, those are all examples of positive feedback. So remember, I said those are unique circumstances. A, childbirth. C, blood clotting. Even D, oxytocin during breastfeeding. Oxytocin is that feel-good hormone that is going to continue, and it helps with the baby and the mother, helps them bond during breastfeeding. That's one of the myriad of benefits of breastfeeding, is that that oxytocin feel-good hormone is being released, and it helps to foster that bond. You guys have been so fantastic today. Well done, you. Let's take a look at what we have here just to wrap up. So I always like to bring the QR codes back up just in case you missed them at the beginning. And obviously, like, I saw a couple comments of people talking about they need to work on like negative and positive feedback. I would highly recommend you go ahead and try both of our question bank and our on-demand videos have free demo. Okay, so you have a free demo time. So you might be able to find some help there. You might be able to find some help on positive and negative feedback there. You can also get some help with it by rewatching old webinars. That would be important as well. Another way to get some help on that. But obviously, if you need some assistance and you just don't think that that's going to get it done, hey, give me a shout out and we'll do some tutoring. I'd love to tutor you. Remember that we do bundle. So we have got our SurePass that puts pretty much everything together except for that 1 hour private tutoring. And if you're thinking you need that 1 hour private tutoring, get that SurePass Pro Pack. And these packages save you a little bit of money by putting them all together. Lastly, there are your tutors. So if you need me for science, scan that middle QR code. If you need Brandon for math or Tammy for English and reading, scan those QR codes. And remember, we have practice questions and little tutorials every week. We have generally right around 2 practice questions and 2 tutorials on our social media platforms. So check out TikTok, Instagram, Facebook, and email us if you need anything at all. Okay. Oh, and it looks like a few people had noted that they bought SurePass. So woohoo, well done. You are certainly going to ace the TEAS with all of those resources. And last but not least, please take a moment, scan this QR code, and let me know how you like today's session. Let me know if there's anything else I can do to better serve you on these free webinars. So I will leave that up for a little bit, but without further ado, I believe we are done. If you are enjoying my webinars, my next one will be macromolecules and cells. It will be the content on June 2nd and the practice test on June 3rd. Those will both start at 3 p.m. Eastern Standard Time. Okay, you guys have a lovely afternoon. I'll leave this webinar feedback up for just a few minutes. You take care.